Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled, The Development of Covalent Drugs with New Emerging Technologies. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for you for future streaming on xtalks.com. At this point, I'd like to thank Aragen Life Sciences who developed the content for this presentation. Aragen Life Sciences is a leading R&D and manufacturing solutions provider for the life sciences industries worldwide. Aragen offers end-to-end integrated or standalone solutions to advance small and large molecule programs from concept to commercialization. Established in 2001, the company operates through a global network of 6 sites with a team of 3,800 plus employees and 450 plus PhDs. Its expertise and experience have enabled over 450 customers, including 7 of the top 10 pharma companies globally, in advancing their research programs from early discovery through development and commercialization. Now it's my pleasure to introduce you to your speaker for today's webinar, and he is Dr. Simon Haydar. He is a Senior Vice President and Head of Integrated Drug Discovery Solutions with Aragen Life Sciences. Simon comes with more than 2 decades of rich experience in leading integrated drug discovery projects. Over the years, he has spearheaded various discovery projects to develop novel oral therapies for different therapy areas. His functional expertise spans across areas such as discovery R&D leadership, licensing due diligence, biotech alliances and partnerships, external R&D activities, and mergers and acquisitions. Now it's my pleasure to pass over the mic and the controls over to Simon. So Simon, when you're ready, you may begin.
Thank you, Sonia. Good morning, good afternoon, and good evening to all my colleagues online, depending on your geography. It's really a pleasure, and I take the opportunity today to describe to you some of the Covalent Therapeutics platform we have at Aragen, which get me first to my first poll question, Sonia.
We're going to start with our poll question right now, but before we begin that, I just want to mention to the audience that you can participate in this poll question as it's done in real time, and it's going to appear on your screen right now, and there it is. So remember, this is done in real time, so please go ahead and cast your vote, and I'll pass it over to Simon to tell you what the question is. Simon?
Thank you, Sonia. How often do you consider development of covalent inhibitor as an alternative modality for your drug discovery of free drug development on your target of interest? Option A is never, option B, rarely, option C, considered one of the mainstream approaches.
Oh, perfect. Thank you so much, Simon. So we have about 20 seconds left. I'll give the audience members to cast their votes. So it looks like we have the majority of our audience have cast their votes. So thank you very much for that. It's truly appreciated. So a few more seconds left as I see there's a couple more people just trying to make their decision now. Please go ahead and cast your vote. Then I will close the polls, get the results with everyone to share with everyone, and then we'll pass it back to Simon. Okay, that looks like it's majority of our audience. Those, thank you very much for participating. I'm now going to close the polls and share the results. Okay, Simon, so here are the results. We have in our audience 5% picked never. Then we have in our audience saying 32% said rarely. And then 64% of our audience members are saying considered one of the mainstream approaches. So thank you everyone for participating in poll question number 1. Now, Simon, before you begin with your presentation, I'm curious to find about your thoughts on the results of that poll question.
Thank you, Sonia. It's actually really in accordance to my understanding and my experience as well.
Mhm.
Given the emergence of new technology and activity we have in the last 5 or 10 years, which I hope to demonstrate today, does support that there is a high interest in this modality, not only in big main pharma, but as well as many biotech for complicated to target protein. So thank you
Wow.
very much for participating. With that, wanted to present to you today the development of covalent drugs with a new emerging technology. And before I move into the science part of it, I would like to take the opportunity and describe to you a little bit more in detail about what differentiates the Aragen integrated drug discovery. So the concept and really the philosophy of operation is to really partner with biotech and pharma and begin to work as in-house drug discovery team. We blur the line between the separation with the drug discovery engine with a partner and at Aragen begin or end. We work in tandem, hand in hand, to develop the strategy, the scientific hypothesis, de-risk the program along the phases from the target validation enablement all the way out in terms of IND packaging. We work with the same sense of ownership and the same mentality of a can-do attitude, because we understand here that there is a high level of investment and high opportunity here to get to a point where we hopefully delivering an asset, an entity for an unmet clinical need for our patient. Lastly, before we move on forward, there's a uniqueness about the integrated drug discovery here at Aragen. We build it up on a platform combining the enablement of Aragen technology along with the structural biology and crystallography we have with Proteomics, from crystallography to cryo-EM, and adding onto it to the computational biology and the data science from our sister company set up for machine learning and AI. With this trifecta platform, we're capable of taking on opportunity from a ideation or target ID validation, going into hit identification, optimization to a preclinical candidate and IND package. The aim and of our effort is really to provide a solutions from end to end, regardless of when the progress of the actual program of the partner, we can enter at any stage of this discovery platform. Okay, so just setting up the stage here, wanted to describe to you, and I'm sure since 60% of you have been working in this field, you're very accustomed or familiar with the challenges, limitation, and hopefully to describe to you what the new technology and opportunity to address for that. So covalent drug, also known as covalent therapeutic inhibitors, referred to as CTIs, could provide pharmacological advantage like prolonged duration of action and enhanced potency over non-covalent drugs. So in recent years, the discovery of covalent drug has been boosted primarily by rational medicinal chemistry design and facilitated actually by emerging a new technology in proteomics, which have helped to identify covalent drug with higher selectivity by identifying their binding site. Now, with this unique mechanistic property that differentiate them from non-covalent inhibitor, covalent drug can be administered at the lower doses and a frequency reducing off-target event and increasing patient compliance. Now, it can be a very effective strategy for low vulnerability target that are high level of occupancy are needed to generate the desired pharmacological outcome. So covalent therapeutic can also be a strategy to overcome some of the drug resistance mutation in the gene encoding of the target protein, like for example in the EGFR target. Now with that comes in significant challenges and limitations that have been identified over the years of discovery in this area. So a key challenge associated with CTI is toxicity. So following an adverse event after administration of strong covalent drug was very difficult to perform or to accomplish. But for days, the drug could no longer, even though it's no longer detected in the systemic circulation, it still can be bound to the target and may elicit AEs. So electrophilic warhead can react with a nucleic residue on protein other than the intended target, such as conjugation with the glutathione, which can lead to off-target effect. This can lead to idiosyncratic toxicity and haptenization, which is an immunogenic response. So, but with the limitation comes opportunities to discharge these risks, such as targeting the non-catalytic amino acid, for example, deploying modern technique in ration design for warhead to fine-tune the reactivity of the electrophile. So recent advances, for example, in a PROTAC covalent approach is another exciting frontier that can be exploited that could provide advantage over both the non-covalent or PROTAC strategy. And I'll show some example of that. So considering the reversible covalent inhibitor to dial in the residence time of the covalent inhibitor on the target site is another approach for mitigating those risks. So these approaches are all within their experience and within reach of Aragen integrated drug discovery. Let me give you a little bit more a perspective on the historical approach, which again, I think many of us or many of you are experienced with that. So covalent inhibitors are compound designed, as I mentioned, to form covalent bond with a specific molecular target. And as mentioned earlier, covalent drugs have pharmacological advantages such as prolonged duration of action and enhanced potency over non-covalent drugs. So if you imagine that despite the introduction of the first covalent drugs such as aspirin, penicillin, or omeprazole, which is the first PPI proton pump inhibitor, there remains skepticism in the pharmaceutical industry for a long period of time to develop covalent inhibitor in the last 50 years. But there's this really a spike that happened around the 2000 when the trend has begin to change. And the graphic here that you can see in the last 20 years, this emergent technology have picked up some steam. And there's over 50 covalent drugs that reach either the market or an advanced clinical trial. And one of the most notable example, for example, in the last state, the Mirati compound for the KRAS G12C, which a few months ago, yet is another exciting application for a covalent therapeutics. Here I'm going to give you a couple examples that actually are pretty exquisite demonstration of the platform. So avibrutinib and zamipritinib are both next generation of Bruton tyrosine kinase BTK inhibitor. So Bruton tyrosine kinase is a kinase downstream of the B cell receptor that is critical for the B cell survival and proliferation. Here's the fun fact. Avibrutinib was originally designed by Celera Genomics in 2005 as a tool compound just to study the biology of BTK. Celera sold that compound in 2006, part of their package deal to Pharmacyclic, along with collaboration with J&J. That molecule became moved into the clinical development and became a clinical asset. So avibrutinib formed a covalent bond with cysteine 481 in the ATP binding domain of BTK. They very rapidly absorbed, eliminated after oral administration within 1 to 2 hours at peak concentration, and initial mean half-life for about 2 to 3 hours in human. The fact that the drug covalently bind to BTK for at least 24 hours provide the opportunity for daily dosing while minimizing the duration of the off-target effect. The next generation compound, zanubrutinib, has also very specific target binding profile to avibrutinib, but it also binds more potently and it shows a fewer off-target effect related to other enzymes such as EGFR or ITK or JAK3. Mentioned to you one of really my favorite example that recently emerged is the KRAS MRT849. So despite decade of research, effort to directly target KRAS have been very challenging. So MRTX849 is identified as a potent, selective, covalent KRAS G12C inhibitor that exhibit favorable drug-like property, selectively modify the mutant cysteine 12 and GTP bound KRAS G12C, and inhibit KRAS-dependent signaling. And there's many, many more examples in late-stage development and also in the late-stage preclinical development on multitude of target in therapeutic area, not just in the oncology side, that take advantage of covalent therapeutic modality. One of the really eye-catching, again, a new frontier, as I mentioned, is the covalent PROTAC. The covalent PROTAC combined its cutting-edge research with area of targeted covalent inhibitors with a proteolysis targeting chimera with PROTAC. So this is a nascent field. However, the research has already demonstrated several interesting findings and holds an immense amount of potential to expand the druggable proteome. So here it comes to Sonia, to my second poll question of the day.
Yes, I'm going to launch it right now, Simon. And there it is, audience members. Go ahead, Simon, with the question.
For those of you who have worked in this area, or those of you who are really learn about this area, what are your major concerns when considering development of covalent inhibitor? Is it yours A, the selectivity, B, toxicity, or C, the ADME properties?
Okay, and now, oh wow, look at that already, Simon. We have more than half our audience have cast their votes, so that's great. For the rest of you, if you could go ahead and do so, just like before, we will give you another 10 seconds to do the simple poll question to answer it, and then I will close the polls and share the results. Okay, that looks like it's the majority of everyone. So thank you very much. Now let's take a look at the results. Okay, so here are the results. In our audience, 32% picked selectivity, while 46% toxicity, while 21% ADME properties. So thank you very much, everyone, for participating in poll question number 2. And now back to Simon.
Thank you again, Sonia. So this is, again, a quite representation of the challenges that we encounter as a medicinal chemist or drug discovery developer taking on a covalent inhibitor as a modality. So the key element here is how we de-risk those elements very early on, so we don't encounter a potential issue that we could terminate or derail the development of our asset down the line. So I wanted to show you, and I will describe to you more in detail, some of the emerging technology and application we have here at Aragen that we have deployed exactly for the reason that you had pointed out in the poll question. So we talk about in terms of the potential design. So we deploy a variety of computational approach in our computational drug design, a suite of software, both is it in-house generated and also combined with the Schrodinger software that take a look at the design of the ligand with appropriate positioning of the warhead. And we modulate that virtually the warhead in order to really fine tune or at least understand the reactivity and the activity of the ligand. We obviously have a knowledge base. There's a significant resources that we have gained at Aragen over the many years working on this field. So that knowledge base is harnessed and is deployed for that purpose. And our relationship and our discovery platform that harness also Proteomics covalent inhibitor screening, we were able to do a wet screening using a technology, either MST or orthogonal conformation with nano-DSF, where we have either in-house or a third-party small combined library of covalent warhead or inhibitor that give us the starting point in the seed that feed in both in the design strategy and the computational approach. Without the ability to do a reagent biology, so this is that momentum cannot be sustained, but we have a very strong reagent biology team that continue to help with providing the right protein structures and the motifs that we need to use for our screening platform, putting together an enabled flow scheme that take us from the ideation through primary assays, be it the biochemical or cellular assay. And I'll just again provide a little bit insight about the technology deployed specifically for covalent therapeutic, and that also linked together with our pharmacology and data visualization and data analysis. So altogether within that platform, capable of putting a very concise, well-connected flow scheme that take on and most novel protein structures into a discovery platform engine, and hopefully that we can culminate in a hit to lead, and eventually our goal is identify a candidate that can move on first in man. So let me talk to you a little bit more about some of the approaches we use, as I mentioned in this building the connected flow scheme. In our computational approach, for recent years, the discovery of covalent drug has been boosted, like I mentioned, by rational design. And I pointed out it's facilitated by technologies such as proteomics, which really enable identification of covalent drug with higher selectivity by finding the specific binding site. Now, traditionally, it's believed that warhead often have to feature an electrophile that attack a cysteine. Now, with a multitude of warhead that has been developed over the last 10 to 20 years, we can right now apply a different target-specific amino acid residue, enabling scientists to dial in the warhead electrophilicity and target specifically other bases besides cysteine, such as tyrosine, lysine, serine, or histidine, or threonine, greatly expanding the opportunity to design better covalent drugs. From a computational perspective, as I pointed out earlier in my conversation, this is a strong platform that is stand on it most of the medicinal chemistry ideation and plan that launched for, especially for area or protein that don't have a precedent, and we're not really using references or publicly available scaffold that we can build on. So we have a multi-step that talks about preparation, first in the term of reaction and covalent docking, that goes into a more selection and minimization of the pose. And finally, it goes into sorting and ranking. In each of the steps, we take on one step forward and identifying the proper modality, the proper confirmation, and the computational approach to give us the best chance trying to delineate the structural motif for which we want to build on the platform. So won't give you in a lot more detail on it, but I would be happy to follow on with more conversation around the computational approach, or even have a separate demonstration to those of you who are interested in this field. All it start and end in really creating a well-connected flow scheme that allow you to address the several key hypotheses, both on the biological targets or on the chemotype. I mentioned in the approach we had at Aragen that we had ability to react from a target validation phase. So often we have an opportunity where we need to understand more the mechanism of action. So through our computational biology, we set the stage where we have a target validation that delineate the computational biology behind their mechanism and feed into a barrier of in vitro and in vivo confirmatory assays. Once we have the cascade biology understood, we start our hit identification phase. And I did say that our hit identification stage has a multi-pronged approach. Partially is due to our machine learning and AI that come in from Accelera, along with the computational support internally at Aragen, that also can have an opportunity to do some screening from our Proteros, or using a very discrete small library of a covalent ligand that we can allow us to begin to understand the opportunity of a new establishing a new chemotype. Then the second stage goes on is setting up in place a battery of assays, both in terms of biochemical or cell assays as well. Understanding the kinetics of a covalent drug is very key and important. As I mentioned before, the activity and pharmacology can be elicited way after the compound is still detected in the systemic circulation. The traditional approach about for your PK/PD relationship is really specially designed for around this type of modality. It's not your atypical traditional small molecule inhibitor PK/PD approach. I'll try to hit on some of that part of it. And then goes into successfully, if we have a tractable hit from a hit expansion, we can delineate the strategy from a hit to lead development using very similar scaffold, the same very similar connected flow scheme, looking for enablement for more drug-like property that hopefully would lead in, would culminate into lead compound. So that's in the first earlier stages of the covalent therapeutic drug development that can last between, in the first phase identification, somewhere around 6 to 9 months. And our lead identification could go up somewhere between 12 to 15 months based on the tractability of the target that we are pursuing. Okay, let me explain a little bit, and my apology if some of this information is pretty available to many of you, but for those of you who have not really delved into this technology before, let me explain to you some of it, what the covalent therapeutic entail. So there's two modes of action for inhibitor of covalent drug. One is the reversible and one called irreversible inhibition. In the first step, the inhibitor, the I, bind to the target protein and form a reversible protein inhibitor complex. That's a PI. The potency of the first step is defined by the binding constant Ki. The Ki term describes the concentration of the inhibitor required for half of the maximum potential rate for covalent bond formation. In the second step, the nucleophile now react with the electrophile of the ligand, forming what we call covalent protein inhibitor complex. That the Kinact is a first order rate constant describing the maximum potential rate of covalent bond formation. So overall, the rate of covalent bond formation from free unbound protein to the covalent protein inhibitor complex PI is defined the ratio of Kinact of Ki. So the Kinact over Ki can be determined in those two steps. In a binding assay, the total occupancy is measured over time at different inhibitor concentration. As I mentioned, these are the kinetics that are very important that can help you understand the potency and how do you really iterate on the structure-activity relationship. So for that, understanding the modality in terms of if a covalent inhibitor is a reversible or irreversible, there's a multiple tools and technology at the fingertips of the scientists. And here I'm describing only a few of the methods that are typically deployed routinely. One, we can really perform a time-dependent incubation. In order to understand, for example, the drug target residence time, jump dilutions is another powerful approach for measuring the residence time using very physiologically relevant conditions. So in the jump dilution experiment, for example, the enzyme and the inhibitor are allowed to form a complex at saturation concentration. Then the complex is then diluted rapidly, and the recovery of the enzyme activity is monitored continuously as the inhibitor dissociates. So the K off is estimated from the rate of activity recovering. So this is also, so you're not only monitoring the kinetics in terms of K on, but K off could also play a very good factor in the design of the experiment for your PK/PD work. Another approach here is the dialysis of protein compound complex is another method to measure the reversibility. One typical other also very frequent method is performing a drug washout from the cell-based assay. So an assessment of duration of action of a drug. All of this will tie back to your in vivo pharmacology efficacy study, as well as you're performing your PK/PD relationship and target engagement. A cellular assay, again, there's the key role or the key objective and mission statement of the biology team here is to put together a disconnected flow scheme. So not only looking at a target binding, but looking at what it's about more of the biological mechanism. And it's often done in a multitude of cell assays representing the disease state or the mechanism of action. From a cell-based reporter assays, it could be done through also a target engagement assays. And we use a different technology and allow us to understand the kinetic and its relative application on the biology. Once again, this is a more thorough, it would deserve another separate conversation just to understand what the technology and when it's really deployed and the readout from those technology as well. So in essence, what I wanted to show today is that we, within the platform technology we have at Aragen, we have designed the tools and resources to address both the in vitro, the cell-based, the mechanistic, multiple platforms can be deployed in the in vitro with a multiple technology. And we have a significant automation, and all is directed to measure. So not only the IC50, which is kind of calculate, show you the potency of your ligand to the target, but also all the kinetic from K on, K off, Kinact to Ki, and all other parameters within the context of how you really interpret the potency you're seeing both in the cellular assay, and hopefully you can translate it mechanistically into more readout in your pharmacology, in your PK/PD relationship. So elements such as biomarker modulation, target engagement, target occupancy becomes a front and center for understanding the dynamic between the cellular, between the target binding and the efficacy readout. So with that, there's a multiple key considerations on it. So we talked about, I think, some of you had alluded, drug safety. So early challenges is how do you really mitigate against idiosyncratic reaction attributed to the reactivity of the electrophile? As I mentioned, because of the reactivity, the target covalent inhibitor can really solicit or induce an immune response on it. So understanding the relationship, and we have, as mentioned, deploying the multiple tools from a computational to innovative way of designing new warhead, it allowed the medicinal chemistry the opportunity to span the spectrum in terms of a dialing affinity for the electrophile to the nucleophile. One of the other mitigation possibility where actually a high potency and high selectivity are the hallmark of potentially proper drug product profile, looking at the CYP-mediated reactive metabolite very early in the discovery stages, it give you a better chance of discharging those risks down the line in the development side. Obviously, those have a very rippling effect on your clinical pharmacology. Ability to be able to understand the kinetics, understanding when you have a drug wash opportunity. So if a pharmacologist administering the drug and have encountered an adverse event, ability to understand when the drug is no longer within the protein site, giving the opportunity to have a recovery. So all those kinetic is really part of mitigating the possibly considering the target protein synthesis and degradation rates also play a factor in deciding on the mechanism of action or the target profile. Non-clinical pharmacology is another area that one has to be very aware and front and center in their strategy. So conjugation of TCI with the glutathione is another way of measuring and looking at it very early. It may become even part of your primary routine screen and as you are dialing in different warhead on your molecule. Protein binding is play another factor as well. Some of the traditional excretion, non-traditional excretion pathway, like binding to albumin, aftinib is an example of that. Measuring it for in vitro evaluation of covalent binding to plasma protein, allowing to understand that relationship. Now, as I mentioned, glutathione conjugation can occur both in the liver and also in the extrahepatic tissues. So predicting it better could also be achieved by considering the whole blood stability and blood binding. So before I bring it into a more of a conclusion, it give me, this is this poll question number 3, Sonia.
Yeah, I'll launch poll question number 3, and there it is. So remember, audience member, this is done in real time. So please go ahead and cast your vote. I'll give you 30 seconds to do this poll question. So Simon, over to you for the question.
So after sitting through today, and I admit I've presented to you at least a hallmark of the platform and technology, are you more willing to consider developing TCIs in your discovery engine? Option A, maybe, B, never, or C, certainly for those of you who already have continued to apply it.
Okay, now let's see. Okay, I'll give just a few more seconds because that looks like the majority of everyone has cast their poll. Looks like there's just a few more people there just waiting to launch it. Just go ahead by selecting any of the 3 answers below. Okay, perfect. Now we're going to close the polls and take a look at the results. Okay, so here are the results, Simon. We got 39% of our audience members saying maybe, and we have 0% saying never, and 61% of our audience saying certainly. So thank you very much, everyone, for participating in our final poll question. Now over to Simon.
Thank you. So at least I'm delighted to see some of you who have never used the technology or have open mind to using it as a potential modality for your discovery engine. So finally, let's see. Oops. Okay, as a summary for Covalent Therapeutics. So in summary, I pointed out to using proteomic and reactive fragment screening that we have to go after targets that were once considered undruggable is really a hotly pursued area of many companies. And the data speak for itself. Over 90% of protein in humans are actually considered undruggable. And however, with this emerging new technology and better understanding the risk and liability and how to mitigate those liability, that allowed the scientists to tackle the area of the proteome, particularly the undruggable proteome, in a way that we couldn't access that before 20 years ago or 10 years ago for that need. So a boom in covalent drugs will continue, and there will be a new innovation almost on a monthly basis, new publication, whether it be new reactive group, expanding library, both commercially or in-house library that are used to screen possible targeting protein previously thought to be undruggable, and going after a target with a covalent therapeutic inhibitor is no longer considered to be a taboo or as much as challenging as it was in the past. For that, I remain for me to thank you very much for your attendance today. Be happy to answer some of your questions today. Back to you, Sonia.
Well, first of all, we'd like to say thank you very much, Simon, for that very engaging presentation, for all that detailed information that you shared with us. So we truly appreciate that. And there he is. He's put on his webcam because he is ready for the Q&A portion of the webinar to begin. And there I am, everyone. Okay, so Simon, while you were speaking, I have a ton of questions that are still coming through. We have a very engaging audience here. So let me start with this first one here. Just a reminder to our audience members, you can continue sending in your questions by using that questions window located on the right-hand side of your screen, and we'll try to attend to your questions during the time that we have together. So our first question here, Simon, is where do you see the future of covalent drug development?
I'm a very strong champion of the area and a very strong believer of the area, having been working in this and seeing on using actually proteomic and reactive fragment screening to go after those targets that were once considered undruggable. As I mentioned to you, is a very hotly pursued area by many companies. In my belief that the boom of covalent drug research is going to continue, and it's going to be continuous innovation in this area. I would venture by saying that it certainly is going to be mainstream for many of the drug development companies, as you consider in different modality, being a PROTAC or being a typical traditional inhibitor, but covalent therapeutic are here to expand and to become a strong workhorse for many of this challenging protein target.
Okay, thank you for that very detailed answer. Let's go to our next question here. Okay, so someone in our audience is asking, how do you choose covalent inhibitors versus PROTACs? Any advantages of PROTACs over conventional covalent inhibitors?
It's a question we actually pause and wrestle with initiation with every single program we have in. Depending if this project if the target is ubiquitinated, or it depends if we can consider there are certain cysteine or amine at certain bases within the reach where in the binding site. The computational support we have is actually give a lot more data-rich packages to allow us to design the strategy accordingly. And sometimes, as I mentioned, is the best of both worlds if we can tackle it with a PROTAC that have at the end terminating with a covalent warhead. The target product profile that is for the asset, for the mechanism we're looking for, is also have a very strong influence in terms of the modality we had choose on. But given that we are beginning to understand how we design a PROTAC in terms of the drug-like property from absorption to having an orally administered drug, we are a better position to really tackle is to stay beyond the rule of 5, so to speak. And I think it's really no longer an intimidating way to go after those large molecular space based on the technology we have in. So I think most of the thing we rely on is really understanding the biology, the computational biology, and understanding the protein structure as quickly as we can. In that way, would really inform us about what the best way of going forward. And most of the time, we do things concurrently to de-risk the program very early and see which of the approach may be more successful than others.
Okay, thank you so much, Simon, for that one. Okay, let's go to our next question. This audience member is asking, hi, he says, does a covalent inhibitor typically need some intrinsic affinity, i.e. non-covalent, in order to get the drug to the site before it reacts? If yes, how potent does it need to be?
Yes. The answer to the first part of it, not always. We've been able to sometimes move on from a target where the traditional inhibitor have a very low potency, and then we've been able to develop the compound with a covalent warhead that have an exquisite potency. Typically, the potency of the inhibitors with a covalent warhead is a pretty, let's say, high potency. The reason why we look for a very potent compound is to mitigate the potential off-target reactivity and off-target selectivity. The higher the potency we have, because we know the residence time of the covalent drug could be several hours. So the minimal amount from a drug burden, drug pill burden that we can administer, the better we have the margin for safety we can work off of it. But again, there is also a modality we didn't talk about one. The reversibility is an important part to mitigate that area. And recently, even we have semi-covalent. People have been working on boron as another potentially semi-covalent warhead that has seen very significant success in mitigating also risk associated with off-target activity.
Okay, thank you very much. I just got so many questions coming through here. I don't want to miss any out. Let me just go to this next one here. Any thoughts on target identification of covalent inhibitor generated from phenotypic screening strategy?
Yes, the phenotypic screening strategy is coming back. My recollection is when I was at Eli Lilly, we have had over 10 years ago, and we abandoned it because deconvoluting the target became an enormous effort and often did not lead to better understanding of the sequence of the mechanism itself. However, again, the computational biology that we have developed the last few years in the platform we have currently at Aragen with our sister company allowed us to go back into this field with more eye-open approach for it. So we have been able to utilize the proteomics very significantly. The technologies allowed us to understand better what are the best way to, what are the best part of the protein to target and how better way to target them. I also would want to point out that from a structural biology, cryo-EM technology has been another tool, another important tool that emerged in the last few years that helped a lot understanding that binding motif that it didn't really had been a prominent 10 years before, and not all the protein that actually could really fall into a optimized condition for crystallography. So the work that we have been doing with Proteras really gave us other opportunity, more shots on target to understand that the approach better than we had before.
Thank you very much for that, Simon. Simon, I'm not sure if I asked this one already, so forgive me if I did. Where do you see the future covalent drug development? Did I ask that already?
Yes, you did.
Okay, thank you very much for that. That seems like it came in twice. All right, let's go to the next question here now. Okay. Okay, so was this one asked as yet? How can we use covalent inhibitor designing strategy for low potency compounds? Can we explore this approach for when we are unable to increase potency further?
Yes, and so for targets that are very low potency, for example, that I say are more featureless, they're where the binding is a much more I'd say, featureless species. By doing some of the scanning for potentially bases, we can really look into areas where we can target the specific bases with the proper warhead. So the actual binding, it does not have to be tight in terms of as a ligand, as long as we have access to some of those bases on the protein. So it gave actually another opportunity for those areas where small inhibitors in terms of some of the pi-pi interaction site has been very difficult, or even becomes maybe outside of druggability, if we can, based on the inhibitor side that we have to develop in order to occupy all the productive interaction that we think we need to do to elicit that potency. So they're usually downstream, the inverse relationship to benefit as we are growing the molecule. With the covalent approach on, you tend to be continue to work within a space of a smaller size, druggable like compound, but having exquisite potency because you have that direct interaction selectivity that you can be built in from the reactivity of the electrophile with the base.
Great, thank you very much, Simon, for that. Let's go to this next question here. Can you comment on orthogonal confirmation assays? Are they used to mitigate off-target binding of TCIs or to confirm the primary assay? It's a very interesting question.
Yes, and I say the answer for this is often both. The reason why I would say in the orthogonal assay that we have used on it, it's very important that we have to, especially in the phenotypic screening that we do, in order to understand that the activity, or at least the primary activity we're seeing it, is directly related to the pharmacology that we would like to elicit. So often, for example, DNA intercalator could really be producing some of the observed potency from the primary assay we have in. So having an orthogonal assay ruling out DNA intercalator could be a very important part in order to choose the right starting point or right scaffolding, especially from a hit screening that you have where you have multiple choices. Again, the idea is to enhance the value proposition where you want to place your bet. So those orthogonal assays are pretty important because it gives you better high level of confidence as you're still building your discovery platform around a scaffold, that the data that you're seeing is really linked directly to the efficacy that you would like to hopefully dial in down the line.
Thank you so much for that detailed answer, Simon. Let's go to our next question now. This audience member is asking, how do you think, or your thoughts, if one could target active site lysine?
Say it again. Could you repeat that question one more time?
How do you think if one could target active site lysine or lysine?
Yeah, lysine. So I think I featured one slide in my presentation where I show you such a diverse warhead design that are available right now as a toolbox for the medicinal chemist and at their fingertip. So there's no shortage of opportunity to continue to innovate in terms of the electrophilic warhead. We're not talking anymore about acrylamide or cyanoacrylamide, which are the hallmark features that you have seen in most of the drugs that are out there as a covalent, like many of the antiviral may have that warhead on. But recently, that the ability to even design what we call masked electrophile, where it reveals itself right at the site of the target. It is really eye-opening and fascinating to see the technology emerging and understanding better. And it's all helped by the computational tools, the software tools that are currently in the available for the scientists that has either from Schrodinger or from other machine learning platform, and including within our own development. So I'm not sure exactly. I mean, it has to be based on an actual protein scout to be able to better understand better answer it. But I remain very certain, given that we have that multitude of options, that none of the most of the target we go after that have an exposed or a retainable lysine within 5 angstrom, for example, and within reach, are druggable, are be able to be approved by a covalent drug. And it's been, and I wouldn't shy away from a protein that have that type of a structural motif in it.
Okay, this looks like it may be our last question. This audience member is asking, are there any approved drugs that feature reactive handles that aren't based on acrylamides?
That's actually a good segue to my earlier response. Indeed, there are already not only drugs in development that are here, there are already drugs that are approved that have electrophilic handle not based on acrylamide. Take, for example, the cancer drug bortezomib, which is actually use a boronic acid to bind to threonine. I mentioned boron is right now is a pretty interesting motif that has been used more readily than I've been having before. It's no longer as just a reagent, but actually it's shown that it could become it's a safe additional variant in a molecule that goes all the way to clinical development. Another drug is the antibiotic phosphomycin that actually have an epoxide that target the cysteine. So that's again back to my earlier response about how you can what are the toolbox of different variants of Warhead that we can be deploying in. While there are not too many of those examples, but I'm seeing more trending that medicinal chemists are becoming more comfortable and less averse to walk down an uncharted path outside the acrylamide, knowledge base. And I think you will be seeing more and more in the next few years, more compounds that have diverse warhead, and where acrylamide becomes the minority rather than a majority of compounds that are developed with a covalent warhead.
So you're looking for the last question here that we can do, and oh, here it is. How do you differentiate reversible versus irreversible binding to cis, sirs, or lys, depend on active site residues?
It's all the way back again to the kinetic and the biochemical assay. And again, hopefully when you have the presentation recorded, you can go back into the slide and take a look at the cell-based assay, and both in the biochemical or cell-free assay and cell-based assay. There's a multiple methodology that can allow you to really distinguish between the two mode of action. And the choice of mode of action, it goes back to your target profile. There's not one way favorable over the others. It's really about de-risking some of the liability. If toxicity and selectivity is a pretty important part, you may look at one. If you're looking at the duration of action or duration of binding between your covalent inhibitor and your target is an important factor, you may consider one modality on it. So again, it's a really important question, an important part, but the multitude of technology and approaches that are deployed very early in your decision tree that allow you to understand that and be able to iterate and do a different ideation around this.
Okay, give me one more second here. Just checking to see if we have any more questions that are coming in from the audience. So give me one moment here just to see. So audience members, remember, you can send in your questions. We do have about a few more minutes left. We can answer probably another one. Just giving the audience a chance to look and see if they have any more questions coming in. Okay, I think that is it. Oh no, we do have one more here. Okay, this audience member is asking, please, your thoughts on covalent agonism and its future.
This is a very provoking question. Conversations on. This is pretty intriguing. Just to be very transparent, I haven't seen yet an opportunity for deploying that for agonism. Often it's been more of a replacing an inhibitor, but nonetheless, even if we are recently been working on PROTAC for a more of a neuroscience target and neuroinflammation, I don't see why we cannot really consider modality in this. So I'll look into it more, and hopefully on the next interaction, I'll be able to address that more in a more educated forum. But I'll keep an eye on this new modality, new approach for it.
Okay. Oh, thank you so much, Simon. I'm sure we all appreciate that. Let's see. I think we're just going to close with that. All right. So thank you, everyone, for participating in today's webinar, and thank you, Simon, for answering all those questions. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Now, if you have any further questions, or if we couldn't answer all your questions, please utilize the email address that's on your screen there, and there's one there that you see that you can reach out to Simon or the team there that will help you with any of your inquiries. So please go ahead and utilize that. Now, and give me a few seconds, I'm going to send you a link in your chat box. You'll be able to view the recording of this event with that link, and also share this link with your colleagues once they register for the recording as well. Now, please join us in thanking our speaker, Simon. I hope you found this webinar informative. I'm sure I did. So Simon, thank you very much for your time and for answering the questions. Okay, it has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you, Simon. Bye, everyone.
Bye-bye.
Bye.